And so the shroud is believed to be a holy relic because you don't get much more holy than the actual blood of Jesus.
The Shroud of Turin has real human blood on it. It has DNA from the Middle East, including markers found among the Druze. The cloth carries the image of a crucified man, encoded in a way no artist and no laboratory has ever reproduced. And for 30 years, a carbon dating test called it a medieval forgery. Both things are true, and neither is resolved. So what is hiding in the threads of the most studied fabric in history? The negative that shouldn't exist. What first pulled this cloth out of faith and into science was a photograph taken in 1898 by an Italian lawyer named Secondo Pia. Pia was allowed to photograph the shroud for the first time, and when he developed his glass negatives, he saw something that stopped him cold. The faint, shadowy stain on the linen reversed into a sharp, detailed, positive image of a man's face and body, complete with wounds. The image on the shroud was behaving like a photographic negative centuries before photography existed. That single discovery turned the shroud from a religious object into a forensic one. Over the following decades, scientists from nearly every discipline aimed their instruments at the cloth. What they found only deepened the mystery. The image sits only on the outermost fibers of the linen, a layer so thin it measures just a few hundred nanometers, barely touching the surface of a single thread. There is no paint, no pigment, no brushstroke, and no binding agent of any kind. In 1978, a team of American scientists called STURP spent over 100 hours examining the shroud directly, using ultraviolet, infrared, X-ray, and chemical analysis. Their conclusion was blunt. The image could not be explained by any known artistic technique, and no one in any laboratory has been able to reproduce it. Whatever created the image, it was not paint. The STURP team included chemists, physicists, and photographers from institutions, including the Air Force Weapons Laboratory and the Jet Propulsion Laboratory. They spent 5 days in direct contact with the cloth, running 33 different tests. Their final report ran to hundreds of pages and reached a conclusion that remains unchallenged 4 decades later. The image is not the product of any known artistic process, and every attempt to reproduce it, from laser experiments to chemical vapor deposition, has failed. There was something else. When researchers converted the image data digitally, they found hidden 3-dimensional information encoded in the shading, as if the image carried the actual distance between the cloth and a real human body. A painting cannot do that. A photograph cannot do that. No medieval forger, working with the tools of the 13th century, could have produced a 2-dimensional image that a modern computer can read as a 3-dimensional map of a human form. The image alone was enough to keep scientists arguing for decades, but it was only the surface of the mystery, because when researchers looked deeper into the blood on the cloth and the dust trapped between its threads, they found evidence that was even harder to explain away. So what did the blood on the shroud actually reveal about the man it wrapped? The blood of a tortured man. The blood revealed a man who had been tortured to death. In 2024, Giulio Fanti, a professor at the University of Padua, published a study in the journal Archives of Hematology that analyzed the bloodstains on the shroud at both the macroscopic and the microscopic level. His findings were specific, and they were disturbing. The blood was not a single type. It fell into 3 distinct categories, each corresponding to a different stage of suffering. The first category was pre-mortem blood, the kind shed while the man was still alive and on the cross. The second was post-mortem blood, leaking from the body as it was taken down and moved. The third was blood serum, the clear fluid that separates from red blood cells after death. Each type left a different mark, and together they traced the sequence of a violent execution. The blood also flowed in 3 directions: vertical for a man hanging upright, at a 45-degree angle for a body being carried, and horizontal for a corpse laid on its side. At the microscopic level, the findings were even more striking. The blood contained nanoparticles of creatinine and ferritin, markers that in modern forensic medicine indicate heavy polytrauma, severe injuries across multiple parts of the body at once. Fanti also identified evidence of microcytic anemia, a condition consistent with severe flagellation around the kidneys, which would have impaired the man's ability to exchange oxygen. The study concluded that this oxygen deprivation forced the heart to work harder until it failed, pointing to a heart attack as the cause of death. This matched the conclusion of an earlier forensic study from the University of Padua's hospital, published in 2017 in the Open Journal of Trauma, which proposed the same cause of death based on the blood evidence alone. That team, led by Matteo Bevilacqua, estimated that roughly 3 pints of blood pooled in the chest cavity from a ruptured heart, a condition called hemopericardium, which is consistent with the post-mortem spear wound described in the Gospel of John. The wounds themselves matched the Gospel accounts of the Crucifixion with forensic precision. Blood-stained marks across the back are consistent with flagellation, the kind delivered by a Roman scourge. Marks on the head align with a crown of thorns. Blood at the wrists and feet corresponds to nail wounds, and a single, large stain on the side of the chest matches the post-mortem spear wound described in the Gospel of John. The blood type was AB, identified in the 1980s by the forensic scientist Baima Balone. So if the blood is real, and it matches the crucifixion account in forensic detail, what does that tell us about who this man was? The DNA passport. It tells us that the cloth has been carrying biological evidence for centuries, and the newest chapter of that evidence is genetic. In the mid-2010s, and again in a more detailed study published in 2026, a team led by the geneticist Gianni Barcaccia at the University of Padua vacuumed microscopic dust from deep inside the weave of the shroud and ran it through next-generation DNA sequencing, a technology capable of reading millions of genetic fragments at once. What came back was not the clean, single-origin profile anyone expected. Instead, the sequencing revealed a mosaic of genetic material from multiple populations and multiple regions of the world. But the dominant signal was unmistakable. A large share of the human DNA traced back to the Near East, including a specific mitochondrial haplogroup labeled H33 that is prevalent in the region and particularly frequent among the Druze, an ethnic group with deep roots in Syria, Lebanon, Jordan, Israel, and Palestine. The Druze share genetic ancestry with Jews, Cypriots, Palestinians, and Syrians, the populations of the exact region where the crucifixion took place. The full breakdown was surprising. Early results suggested that more than half of the detectable human DNA traced back to Near Eastern populations, while close to 40% pointed toward India, with only a small fraction linked to Europe. For a cloth that has physically sat in France and Italy for centuries, that European percentage should have been dominant. Instead, it barely registered. The genetic signature reads less like a European artifact and more like an object that passed through the ancient trade routes of the Mediterranean and the Near East. The DNA also carried traces of something else entirely. Microorganisms that thrive only in extremely saline environments, the kind found near the Dead Sea, turned up in the sequencing. These are not organisms you would expect on a piece of cloth that had spent its entire life in the cathedrals of France and Italy. They are organisms that suggest the cloth, at some point in its history, spent time in the environment of the Middle East. Barcaccia was careful about what this meant. The DNA, he explained, proved that individuals from wildly different ethnic backgrounds had come into contact with the cloth, either directly in Europe or somewhere along its journey. The cloth reads less like a relic that sat quietly in one cathedral and more like a passport stamped across half the ancient world. None of this proves the cloth is the burial shroud of Jesus. A medieval cloth brought back from the Middle East by Crusaders could carry similar traces, but it does place the cloth, biologically, in the region where the man on it was supposed to have lived and died. So if the DNA points east, why did one famous test point straight at the Middle Ages? The test that almost killed it, because in 1988, 3 of the most respected radiocarbon laboratories in the world, Oxford, Zurich, and Arizona, tested a small sample of the shroud and concluded that the linen was made between 1260 and 1390 AD. The results were published in the journal Nature, and the headline was unambiguous. The shroud was medieval. For 30 years, that finding was treated as definitive, the single fact that closed the case. Then, the case began to reopen. The first crack came from a chemist named Raymond Rogers, who had been the head of chemical research at Los Alamos National Laboratory and a member of the original STIRP team. Rogers examined threads from the same corner that had been sampled for the 1988 test and found that they were chemically different from the rest of the shroud. They contained cotton fibers interwoven with linen, traces of a medieval dye, and vanillin, a compound that disappears from linen over centuries. His conclusion was blunt. The sample tested in 1988 was from a repaired corner, not the original cloth. The worst possible sample for carbon dating had been taken. Rogers was not a fringe researcher challenging the establishment. He had been a department head at one of the most prestigious scientific institutions in the United States, and his analysis was published posthumously in the peer-reviewed journal Thermochimica Acta in 2005. He identified cotton fibers interwoven with the linen, a vanillin coating consistent with medieval dye, and chemical signatures that distinguished the sampled corner from the main body of the cloth. In 2019, a statistical reassessment published in the journal Archaeometry reanalyzed the raw data from the 1988 test and found that the 12 subsamples were not homogeneous. The data were statistically inconsistent, meaning the results could not be trusted as a single measurement. Then, in 2022, a study in the journal Heritage used a technique called wide-angle X-ray scattering to measure the structural degradation of the linen's cellulose and concluded that the cloth was approximately 2,000 years old, consistent with a linen sample from the siege of Masada, dated to between 55 and 74 AD. But here is where the honest part matters, because the other side has evidence too. In 2026, a study published in Nature performed a textile analysis of the fragments from the 1988 test and found no evidence of repair, concluding that the samples were consistent with the main body of the shroud. The medieval date, they argued, stands. So the dating debate is not resolved. It is 2 peer-reviewed studies saying the cloth is ancient, 1 peer-reviewed study saying it is medieval, and a growing body of researchers arguing that the 1988 test was compromised. Should the Vatican allow a new round of radiocarbon testing, or has the 1988 result already done too much damage to undo? The face cloth that matches. There is one more piece of evidence, and it does not come from the shroud itself. It comes from a completely separate cloth, kept in a completely separate city with a completely separate history. The sudarium of Oviedo is a smaller face cloth held in the Cathedral of Oviedo in northern Spain. It has no image on it, but according to tradition, it was the cloth placed over the face of Jesus after he died, and it was carried away from Jerusalem before the shroud, traveling through North Africa and Spain before arriving in Oviedo by the 7th century. The 2 cloths have never been physically connected in their documented histories. They traveled different routes, were kept in different places, and were venerated by different communities. And yet, when scientists compared them, the overlaps were striking. The blood type on the sudarium is AB. the same rare type found on the shroud. The stain patterns on the face cloth align with the blood marks on the face of the man in the shroud image. The pollen trapped in both cloths includes species indigenous to the same region of the Near East. The geometry of the face, measured from both cloths independently, matches. The measurements were made by separate research teams working with separate samples, and neither team knew the other's results until after their own work was complete. The bloodstain patterns on the sudarium align with the wounds visible on the shroud image, particularly the marks consistent with a crown of thorns and the blood flow from the nose and mouth that would accompany a crucified man who had been taken down from a cross and laid face up. None of this is proof. Two cloths with the same blood type and the same pollen could have been created in the same medieval workshop or in the same region of the ancient Middle East. But the independent histories of the two objects make a shared origin difficult to explain. The sudarium was in Spain by the 7th century, centuries before the shroud's first documented appearance in France in the 14th century. The sudarium has been in Oviedo since at least the 7th century, documented in church records and mentioned by pilgrims who traveled to see it. That means it was in Spain, with its own chain of custody, for at least 600 years before the shroud appeared anywhere in the historical record. If the two cloths were forged together, the forgers had to plant one in a Spanish cathedral and then wait centuries to produce the other. If both are medieval forgeries, they are forgeries that were coordinated across hundreds of miles and hundreds of years without any evidence of coordination. The simpler explanation is that both cloths touched the same person. But simpler is not the same as proven. So what does the institution at the center of all this, the Vatican, actually believe? What the Vatican actually says? The honest answer is that the Vatican has no official position. The Catholic Church has never ruled on whether the shroud is the actual burial cloth of Jesus Christ, and it has never declared it a relic. The Holy See took custody of the shroud in 1983, when the former owner, the Savoy family, willed it to the Church, and since then, every pope who has spoken about it has been careful with words. Pope John Paul II called it a mirror of the gospel. Pope Benedict XVI called it an icon. Pope Francis, when he venerated it publicly, used the same word, icon, a deliberate choice that stops short of saying relic. An icon is something that points toward a truth. A relic is a piece of that truth itself. This is not evasion. It is a recognition that the question of the shroud's authenticity sits at a boundary that science alone cannot cross. The evidence on the cloth is real. The blood is real human blood, type AB, carrying the chemical signature of severe trauma. The DNA is real, and it places the cloth in the Near East. The image is real, and no one can explain how it was made. The dating is contested, with peer-reviewed studies pointing in opposite directions. All of this is documented, published, and verifiable. The same data that shows real blood also shows that the cloth carries the genetic fingerprints of the ancient Middle East. The same endogamy that preserved the image also allowed local DNA to accumulate over centuries. The science supports both the extraordinary and the mundane, and the shroud sits at the exact intersection of those two categories, which is precisely why it has fascinated scientists and believers alike for more than a century. But no test, however sophisticated, can answer the one question that every viewer of this video actually wants answered. Whose blood is it? Whose image is encoded on those fibers? Science can tell you that the man on the cloth was crucified, tortured, and buried in a manner consistent with the gospel accounts. Science can tell you that the cloth traveled through the Middle East. Science cannot tell you that the man was Jesus of Nazareth, because identity is not something you can measure with a mass spectrometer. This is why the Vatican stays quiet. The shroud is too well evidenced to dismiss as a simple forgery, and too incomplete to confirm as the burial cloth of Christ. It sits at the exact point where the tools of science run out and the eyes of faith begin. And that, perhaps, is the most honest thing about it. When you look at the face on that cloth, you are looking at a genuine mystery, one that neither the laboratory nor the cathedral has been able to close. So is the Shroud of Turin the burial cloth of Jesus, or the most sophisticated puzzle in the history of science? The Shroud of Turin has real blood from a tortured man, real DNA from the Middle East, and an image that no one can reproduce. It also has a carbon date that says medieval, and a growing body of science that says ancient. The Vatican calls it an icon, not a relic. It is too well evidenced to dismiss and too incomplete to confirm. Is it the burial cloth of Christ, or the greatest unsolved mystery in science? What would it take to settle it?